Hi everyone, thank you for coming. We will start in a little bit. Hi Kirko, come up. Haven't talked with you in a while. How are you?
I'm doing well. You know what I'm saying? I finished up training, so I've been trying to acclimate back to civilian life. You know what I'm saying?
Oh,
So
what
kind
did
of been.
you do? Training?
Yeah, I was in military training like
Oh
since I
wow.
met you until like about a month ago.
Oh wow. That's
Yeah,
something. How did
and
it go?
it went well. You know what I'm saying? I got what I needed to get, so now I'm like back, where I live and trying to get all my certifications set so I can start making adult money. You know what I'm saying? That's kind of actually why I haven't been like around too, because um, a lot of it was like exams and stuff I had to study for, and then since I've been back, getting to hang around the family and doing all this paperwork stuff like that. So I've just been kind of busy, so that's why I haven't been like in a lot of rooms. But I'm
Yeah,
back.
then I'm glad. Well, I guess congratulations to passing all the stuff
Thank you.
and doing that. It must have been hard, so chapeau. I can't imagine. It must be really hard. Hi, Frank. How are you today? Nice seeing you. Hi, Brian. Hi, Eric. Nice seeing you all here.
Hey, Katrina. Hi, Kirko. It's been a while, yeah, but looking forward to this talk. It's a green material, and I'm looking at the slides. I'm trying to, get a preview. So how are things going, Katrina, and all the friends here?
Good, good. Thank you. Busy but good. So, uh, yeah. And apparently, Kiko was very busy, and yeah. So we've all been. You were here. Was it
Oh
last?
yeah,
Yeah, you
yes,
were
yes.
here
I
last
was
week.
I was here, yeah, in the
Yeah,
room.
that was good. So yeah, hi everyone, and nice seeing everyone. If you think this is interesting, feel free to share the room. It will be really interesting. I shared some further reading. Jonathan, thank you. Well, you always do that. That's nice. So, some open access paper if people want to check it out that Matthew shared with me earlier today. Hi, Matthew. How are you? Welcome. So, to unmute so we can hear you, it's all the way on the bottom right.
Sorry, yeah.
Perfect.
Thank you. It's very nice to be here. Can you hear me
Yep,
now?
we can hear you. Welcome. Thank you for coming.
Oh, no worries.
I
It's a
shared in
pleasure.
the chat the papers, and on top is the presentation. If you want to check it out, if it works well, everyone, before we start, feel free to do so. Hi, Bill. I haven't seen you in a while. How are you, Bill? Come up. I'm sorry, Matthew. I was just
Oh,
seeing people in the audience.
no worries. So, do you have a regular crew that always shows up for this, or how does it work? This is my first time at Science Society.
Yeah, we have so Brian, like Kyoko, he went through, he can tell you. So he wasn't here for a little bit, but usually, yeah, he would come, and Frank comes really a lot of times and
Cool.
helps moderate. And then in the audience you see Eric, and Brian comes all the time, and whenever he misses rooms he apologizes. He doesn't need to do. Oh, but and Bill, he was away for a little bit because he was traveling and not having reliable internet. Jonathan, he is always nice and supportive. Yeah, so
Very nice.
yeah, we have more people that come back. It depends on the time of the day.
Yes, of
Uh,
course.
if it's more people from Europe, um, it's more in the earlier rooms, and then
Right. I guess we're missing them right now.
Yeah,
except the
exactly.
night owls maybe, but
But they can listen to the replay, and the cool
that's
thing
true. Yeah.
to do it on Clubhouse is that they will see the room the exact same way we are seeing it. Like they can click to the chat and
Ah, cool.
on the presentation. Just they cannot actively participate. But other than that, they see everyone here. They can click the links and everything. So
Very nice.
yeah. I'm glad you did this. You did the account and came here. It's really nice of you.
Yeah, no, I'm happy. It's always nice to to talk about your science, and to reach a new community is always kind of fun. And I really encourage people to ask questions, because maybe you think of something that we haven't quite thought of yet, and or see it in a different way. I think that's kind of the great thing about science, and especially our science, which is very cross disciplinary. That different perspectives always are always welcome and helpful for us to kind of think about our problems.
Yeah, I agree. That's really the cool thing about being on Clubhouse is people from so many different knowledge backgrounds that
Hmm.
the conversations can get really interesting if people are open to it. So yeah, I agree. That's what kind of made me become addicted to Club was these really interesting conversations that you don't have in conferences.
Yeah.
Yeah, so
Yeah,
sometimes
cool.
it takes a little bit of time to get them started conversations like this. Sometimes in the beginning people are kind of, like scared of talking when
Hmm.
it's a subject that they are not comfortable in. But I think we passed that.
Yeah.
We became all very bold.
Yeah. Oh, that's good.
Over time. So I
That's
think
fantastic. Yeah. Well, I mean, I'm open, so feel free also as I'm speaking to shout out something. I'll try to keep an eye on the comments, in case there's some questions that come up during the talk. So yeah, I definitely am open to also being interrupted.
great. And I
Yeah.
think it's time to start. People will continue to come in, but we start with the introduction and then um, we'll go from there. So yeah, welcome everyone to Science Society. And of course, a special welcome to you, Matthew. And before we start, let me give a short introduction so people know. So yeah, just people know, something more about you.
Hmm.
And then usually we ask like a couple of interview questions if that's okay with you, and then it's time for your really cool research. So
Absolutely.
perfect. So Professor Matthew Harrington, he's an associate professor and Canada Research Chair in green chemistry in the Department of Chemistry at the McGill University. And he's also the co-director of the McGill Institute of Advanced Materials. And he did his PhD at the University of California, Santa Barbara, in the lab of Herbert White, and I hope I'm saying his name right. And he he then did a postdoctoral fellowship, Humboldt postdoctoral fellowship at the Max Planck Institute of Colloids and Interfaces in the Department of Biomaterials, where he then was a research group leader until 2007. And his research is about understanding biochemical structure property relationships and from the function and the formation of biological materials and applying those to design principles for the development and sustainable production of bio-inspired materials, which is really interesting. And I think it's a really great research area to be in. So welcome. And as I said, we usually ask like a couple of interview questions so people get to know you as a scientist a little bit better.
Sure.
So, was it always something you wanted to do, like become a scientist? Or was it just something, you read the book, you had a great teacher, or you just slid, was just by chance? I think it's
Mm,
really interesting for us to learn about the different career paths.
yeah, no, that's a good question. It's funny. I was at my sister's wedding last weekend, and saw a bunch of my relatives who I haven't seen in a while, and my aunt, who used to babysit me as a kid, was talking about how as a kid I never shut up. I never stopped asking questions, even as like a really young guy. Just why all the time, wanting to know everything. And so I think it was always in me to just need to know how things worked. And in particular, I was always interested really in biology. I remember also my nana's house, they had this book on the human body and human biology, a college textbook, which I was just fascinated with as a kid. So I think I always knew I wanted to be a scientist. But in this particular field of bio-inspired materials, kind of, I guess, happened by chance. And it's a very interdisciplinary field that kind of requires background in biology and chemistry and material science and engineering and all these aspects. And I don't know, I feel like somehow I was led here through the different topics that I became interested in. But once I got into the field, it was a completely natural fit. And yeah, so I can say as a kid, I always definitely wanted to be, or I don't know, I was meant somehow to be a scientist, but to actually make it into this field also felt very natural as well.
Yeah, that's really interesting. That it was always something you wanted to do and kind of your passion. So, um, we keep hearing that, and it's really, and I get feedback from audience that are not in scientific fields, that it's very inspiring for
Mm,
them to hear that, and they feel like very positive, like they have more hope that people working on like real humanity problems that are
yeah.
doing that out of that reason. So I always appreciate hearing those stories. And you kind of answered already the second question about, how you went in that field. So
Right.
how did, you know, this, I wrote you about this paper, but you were also presenting other projects. But how did was it hard to get like funding? Did people, sometimes we have people that come here and say, yeah, people thought I'm crazy.
Yeah.
Or they say, oh, this was so easy for this project, I got money right away.
Yeah.
Or it was like some mistake happened
Yeah.
and they thought it was a mistake result and then it became a project. Like, is there some story like that maybe behind?
Yeah, I mean, for the mistletoe. So, I mean, during my PhD, I was working really on the muscle byssus, which I'll talk about in a bit, which is a underwater adhesive that marine mussels use to attach onto rocks at the seashore. And that was in Santa Barbara, where we had the ocean. And then I moved to Germany, and I was lucky enough to work at the Max Planck Institute, which, for those who don't know, is a kind of a publicly funded, government funded research institute that is all around Germany. So they have different topics. So I was at the Department of Biomaterials. And this is really kind of like a wonderland for a scientist in the sense that it's really, so it's not a university, you don't have the teaching requirements, but you're able to just really focus on your research. And they're very well funded. So as a group leader there, I could kind of have some freedom to, I had my own funding that I got some funding to work on specific projects, But the whole goal of the Max Planck is fundamental research. And so they allow you to kind of take risks. It allows you to be able to do that kind of research that if you're constantly, trying to get funding for this or that, you might say, well, this isn't really safe. I probably shouldn't. I probably should go with something safer. But being in this environment, right? I had my safe projects, I had my funded projects, but I had a little extra money that I could work on some risky projects. And so the mistletoe, when it started, was kind of a risky project, and it does have a pretty kind of funny story. So I was trying to learn German when I was there, and I started watching this show, which Katharina, you might know. It's called Sendung mit der Maus. It's like the German equivalent of
Yes.
Sesame Street for North Americans, right? And I was watching the show, and again, I was just watching it to try to improve my German. And I had never seen mistletoe because it's kind of a European species until I'd gone to Europe. So I kind of knew what it was, and I knew it was these things that grew on the trees in the winter that were green all year long while the trees lost their leaves, and I didn't know much about it. And watching the show, though, I saw that it was these berries were a food that birds ate in the winter, and then they would, for lack of a better word, they'd poop out the seeds. But when the seeds came out, they would turn into these long fibers, like really long, sticky fibers that they would then wrap around the branches of the tree that the bird's sitting on, and would start um, growing. The seeds, actually, it's a parasite, so it grows into the branches of the tree, which was just wild. So I'm watching the show, and I'm like thinking to myself, oh my gosh, I have to study this. Like, how does that work? And I looked up immediately any paper I could find on the topic, and there was basically about 2 or 3 papers only that had started to look into how the fiber forms, how the adhesive works. Um, so I was hooked on that, and then I was like, all right, how do I get some of this? And it was kind of close to Christmas time, and at the Christmas markets they had all these. You could go and just buy the mistletoe. So I picked some up, and we were just kind of at home playing around with it. And all of a sudden, I'm in my kitchen playing with these berries and pulling these fibers and hatching my plan to make this my project. And that was probably, I don't know, probably 6 years ago. And it's just been so much fun, working on that and actually getting to work with some amazing students on this project as well. So it was one of those things that was kind of just it was a whim while I was trying to learn German. And I just was totally hooked on it. And you'll see when I start talking about the other systems, there are a lot of similarities between, the different systems I'm talking about. So mistletoe, but also mussels, and then this other weird creature called a velvet worm that I'll talk about as well a little later.
That's a very cool story. And, when I moved as a kid after second grade to Germany, though, I learned German mostly with those shows and
Yeah.
with this cassettes that had the stories,
Mm-hmm.
Benjamin Blümchen and
Yep.
TKKG. Like, I
Yeah.
mostly learned German through those things.
Yeah.
Like, oh yeah,
It's yeah, it's great actually. That show is really fantastic.
yeah. So thank you for giving us this really cool background story. And yeah, the stage is yours for your presentation. And yeah, we are all looking forward to learn more. Thank you.
Great, fantastic. Yeah. So again, Katerina, thank you so much for the invitation to be here with you tonight, and thank you to all the people who showed up and took a little bit out of their time to hear about our science. Yeah, so I think as it might be clear now, our area of research is what's known as bio-inspired materials, biomimetic materials, and that sort of thing. So if you're following along on the slides that I kind of uploaded, this first slide is really it's a paper that came out that kind of asks the question, like, what can we learn from nature? What can we learn from natural materials? and how can we learn from that? And I've underlined a line there in the abstract that says a thorough analysis of structure function relationships in natural tissues must precede the engineering of new bio-inspired materials. And I think that's a good line that kind of says what we're all about. So the premise here is that through evolution, nature has come up with a lot of good solutions for common engineering problems, right? They've kind of through evolution have stumbled upon materials that actually have properties that can outperform in certain ways any of the best engineered composites or polymers or adhesives or whatever. And so that's because it's just billions of years of evolution have kind of stumbled upon these solutions to problems. And the other benefit that comes with nature, and this is one of the things that really motivates our group, is that these materials are inherently bio-friendly. They're inherently recyclable and biodegradable, and they're inherently, therefore, sustainable. So some of the materials I'm going to talk about today are what you would call biopolymers. They're the biological equivalent of a plastic, like a plastic fiber or something like that, or a polymer glue. And inherent in these, because they're bio based, they're based on proteins or they're based on sugar chains like cellulose. They're gonna be more sustainable, right? And so what we think is that we can learn from these systems by if we take kind of a chemist's point of view, we can treat these instead of saying like, oh, that's a biological material. We say, okay, that is a biopolymer. That's like a plastic. And I'm going to try to reverse engineer that. I'm going to try to figure out how it works. I'm going to try to figure out why this works the way it does. And then what we do is we get what we call design principles. So, fundamental physical and chemical principles of that describe how this works. And when we do that, we can then go to the lab bench, and because these are chemical and physical principles, we can copy that and we can try to make new polymers and new plastics or new adhesives. So if I go to the second slide that I have on there, that's kind of our approach. What we do is we start with the biology. We find an interesting model system that has an interesting property. Typically, we aim towards materials that are outside of the body, that are made and function outside of the body. You can imagine something like spider silk, right? Spider silk is non-living. It's a fiber that's produced outside of the body of the spider. So it's there's no living cells. We don't have to worry about all the complication that comes with living cells. This is basically just a plastic-like material. And if you look at spider silk as an example, spider silk is, I'll argue, better than any natural or any synthetic polymer that humans have come up with. It's tougher than Kevlar, and Kevlar is what you use to make a bulletproof vest, right? In terms of toughness, it's 3 times tougher, and it's got really excellent properties. But it's made under bio-friendly conditions. And so the materials that we study, which are the mussel byssus, the mistletoe Viscin fibers that I mentioned, and the velvet worm slime, these are all, let's say, kind of similar to spider silk in the sense that they're made from biomolecules like proteins and polysaccharides. They function outside of the body. They're fabricated in very quickly outside of the body. And because of that, we can study them. Like I said, they're kind of like a plastic that we're going to try to reverse engineer. And so that's our approach. We really take this multi-scale scientific approach where we use methods from biochemistry, from chemistry, from material science to distill and elucidate these design principles by looking across length scales from the biomolecular level up to the higher order organization. And once we've kind of distilled these principles down to their very basic chemical and physical nature, that's when we can say, okay, now we know how this works. Now we can go to the lab and we can try to do this synthetically and see if we can recreate or mimic some of these properties. And yeah, we've been doing that for quite a long time. I've been working on the muscle byssus for almost 20 years. The mistletoe fibers and the velvet worm slime are more recent, say 6 years that we've been working on them. But even in that short time, we've got some really cool insights. And that's what I'll kind of tell you a little bit about today. And with these insights, either our group or with our collaborators, we, like I said, we try to mimic this synthetically to make materials that have what we call stimuli responsive properties. So they can change their properties depending on the local environment, materials that are self-healing, that can be damaged and then spontaneously recover to their native state, and so on. So that's kind of like the overview of our approach. And so with the rest of the time I just wanted to kind of briefly go through these systems. And if
Matthew.
any of them, oh, sorry.
Yeah, Matthew, quickly. So, sorry for to interrupt. The
No problem.
quick clarification. You mentioned the spider silk as well. So,
Yep.
but the slides number 2 is that you work on between the two.
Sorry, I think I lost the audio for a minute there.
Yeah, Frank, you're breaking up in between.
Oh, oops, sorry. So how do I sound now?
Yeah.
Good.
Okay, so yeah, so I was wondering the difference and the similarity between spider silk and the muscle, that because you mentioned
Oh,
spider
yeah, sure.
at the very
Yeah.
beginning, so it seems that the one is much longer. It's actually quite intriguing to learn that the muscle actually has this silk like
Yeah.
mechanism as well. Yeah,
Yeah.
terrific. Thank you.
So that's a great question. And spider silk is one of those. It's kind of the poster child for for bio-inspired or biological polymer materials, right? It's been studied for the longest and probably the most is known about it. Um, Muscle byssus is a different material for sure. It has very different properties. It is also quite tough. But you have to remember, this is also functioning in the ocean. It's functioning in a marine environment. So it has a different set of, um, let's say, needs. The function of this actually is completely, spider silk is evolved to capture insects which are flying. It needs to basically dissipate the energy, the kinetic energy of the flying insect. So it needs to be quite tough and just damping. Byssus also needs to be designed to be damping, because what the byssus does is when a crashing wave comes, those fibers are the only thing that keeps that muscle from being ripped off the rocks. And these waves are really crashing with incredible, speed and acceleration, and then the muscle is experiencing this, right? So they also need to be incredibly tough. So these fibers, when you look at the numbers in terms of the mechanical properties, they're also incredibly tough. They're as tough as Kevlar. So they're about 3 times less tough than spider silk. But they have other properties that are really interesting. For example, well, I might as well go to the next slide. So that's slide 3. They actually have a property that really is what got a lot of interest of scientists early on. They are capable of underwater adhesion. Okay, so imagine that you're trying to glue or tape onto a surface that's wet, or even just maybe it's a humid day, and there's some humidity that's accumulated on your surface, and you want to put some tape on there. You're gonna have a really hard time, because water will create a stronger interaction with that surface, and your tape or your glue can't displace the water, right? So that's going to inhibit you from being able um, to glue onto that surface. Yet this organism, this muscle, has evolved a strategy, a chemical strategy, to be able to glue underwater. So that's one big difference between spider silk. I mean, there are adhesives involved with building a spider web, but they're not typically functioning in a wet environment like this. So a lot of people got really interested in understanding that underwater adhesive, and that was the lab that I worked in for my PhD was one of the really big labs that did that. And what's really interesting there is that they they did back in the 80s and the 90s, they basically learned that, okay, this is made of protein, and now we have to extract the proteins that make up this glue, this adhesive, and they extracted them, and then they learned that the chemistry of these proteins was very different from most proteins. The animal was actually modifying the chemistry of the glue proteins after they'd already been made in a very particular way. And what they were doing is they were adding this chemical functional group that we call DOPA. So it's called dihydroxyphenylalanine. We call it DOPA. And this is a very, if you know anything about proteins, well, there's 20 amino acids, there's 20 standard amino acids. So the muscle was actually saying, well, those 20 aren't working for us. We need this other one because it has to do this special thing. And so they make an enzyme that modifies that protein and adds on this DOPA. And right when I was getting started with my PhD, they just made this really big discovery that the DOPA was actually what allows the glue to stick on wet surfaces. In fact, it was the DOPA that is so good at sticking on surfaces that it just like basically knocks the water out of the way, and kind of takes over. And so that discovery was kind of where I was starting with. And also then they found DOPA in all other places in the Bissell threads, not only at the adhesive surface, but also DOPA turned out to be really good at forming these special types of crosslinks, which were based on metals. And there was all these really neat discoveries around this. So then the engineers kind of figured this out, or they were reading these papers, and they started saying, well, can we use DOPA? Because basically DOPA, the chemical group on DOPA is what's called a catechol. And yeah, these are readily available, and it's actually, DOPA has a similar structure to the dopamine that's in your the neurotransmitter that's in your brain, right? It's a very common and relatively simple chemical structure. And so what the chemical engineers and material scientists started doing, they took this chemistry and they said, okay, can we make underwater adhesives? Can we make coatings? Can we make this? And it turns out that by using this chemistry, this chemical group, you can totally do that. And so that, for example, there's a group out in UC Berkeley, a group of Phil Messersmith, that are developing surgical adhesives that are based on this chemistry, this very simple chemistry that was discovered in the muscle. And they're using these, I think they're in clinical stages now, but they're using them in kind of prenatal surgeries where you can't use sutures. They use this to kind of seal the amniotic sac after the surgery, which reduces the likelihood of premature birth. So again, this was kind of the original research was just kind of a fundamental investigation. That wasn't the intention leading into this to just say, okay, can we make a surgical adhesive? It was really fundamental research out of interest, out of curiosity, to say, how the hell does the muscle do this? And then, it was a lot of biochemistry, it was a lot of material science, it was a lot of, general basic chemistry, physical chemistry, and they kind of, made this big discovery. I don't know if that kind of answers your question. It was kind of a roundabout way to get here, but that's definitely one of the big differences that that we see with mussels versus spiders.
Well, thanks for that thorough, and I guess L-DOPA is
Yeah.
the main. Yeah,
Well,
okay, great.
it is. Yeah, it is very important. There are other aspects to this. I mean, I could, I don't have all. I was told I have about 10 minutes per material, but I could easily talk about the business, for 2 or 3 hours. But I won't do that because I probably have to go to bed at some point. But the other thing we're really interested in with the mussel business, I will say, is the way that it's formed. Because again, like I said, the fiber itself has properties that are as good as Kevlar, but Kevlar is a material that's produced, from petroleum-based precursors. It needs to be dissolved in almost pure sulfuric acid. To get it to dissolve, you have to heat it 80 degrees Celsius. So it's not a very sustainable process. But yet these Bissell thread fibers are produced in just minutes as a secretion by the mussel, kind of similar to the way spider silk is formed. And the way that the mussel does this is that it pre-packages all of the precursors, so these are the proteins, in these tiny little vesicles. So these are kind of small little bags, for lack of a better word, bags or sacks of these proteins. But the proteins, obviously, if they're going to form a material, they have to be a liquid form, right? They have to be able to flow and coalesce and be formed into this material of a certain shape. And so what happens is that the mussel is storing them in this liquid phase, and then they get secreted, and then they have to undergo this really fast liquid to solid transition. And that's what we've been studying like in the last 5 years. That's been really cool to us. And we think that understanding this could provide some information about how to make plastics more sustainable. Or in a more sustainable way. And so the one trick that the mussel, I'll just tell one of the tricks the mussel uses, at least for this Kevlar type part, which is this fibrous core, it actually stores the proteins in what's called a liquid crystal phase. You probably know liquid crystals from your television, LCD, LC display. Liquid crystal display is what it is. These are also liquid crystals like that. And what does that mean? A liquid crystal is something, sounds fancy. It's um, kind of simple in the sense that it's a phase of matter that is somewhere between a crystalline solid, and a liquid. Crystalline solid means that all the molecules or atoms in your material are organized in a very periodic way in the material. So the building blocks, whatever they are, the molecules, in this case proteins, are organized, right? So they have some organization already. But they also are liquid in the sense that they have fluid-like properties. They flow. And so if you have these two properties together, what does it allow you to do? It allows you to kind of pre-organize your building blocks, so they already have this structural order in the fluid phase. And then as you transition them into a solid, it means you can go really quickly to trigger a transition into a solid that has a lot of structure, that has a lot of structural organization. And it's very well known from plastics and other materials that the more structural organization you have, typically with these kind of plastics, the better the properties are going to be. So this is a kind of way that the mussel hedges its bets in order to really quickly create a very highly organized and material that has amazing properties. So that's kind of what we have been getting up to with the mussel. I'm going to quickly switch gears to our mistletoe work, if that's okay. And so I told you a little bit about the mistletoe story when I was talking about the motivate or why I got into this research. And so if you are following the slides, you can go to slide 5, and you can actually see a picture of a bird that's passing some of these berries out its back end, and you can actually see, if you look really closely, there's these string. It's basically a string of these seeds. It's like a pearl necklace almost, where the seeds are all attached to one another on a single fiber. Now, the crazy thing about this fiber, it comes from this tiny little tissue surrounding the um, the seed, which is called the Viscin cell bundle. This thing is 5 millimeters long. Okay, it's like half a centimeter long. You can take this tissue with your bare hands. You can take the Viscin cell bundle, and you can just pull your arms apart as far as they go, and you'll make a fiber that long. Okay, and it's sticky, but as it dries, it loses its tack. But you can draw a fiber from this tissue that's 5 millimeters long. You can make a fiber that's 2 meters long. And these fibers, once they dry, also have an incredible stiffness. They're let me think, 3 or 4 times stiffer than nylon. Okay, nylon, what you do, what you go fishing with your fishing line, it's going to be nylon. These are stiffer than that. They're flexible. They have amazing properties. But if it happens to be humid, they will then immediately pull up humidity from the atmosphere, and they'll become a lot softer, and they can flow again, and they can be pulled. So it's a really interesting material because it can be formed into fibers. It actually is quite adhesive, and it sticks to almost any surface, including metals and plastics and glass and wood. It sticks to human skin. It sticks to cartilage. But because of the nature of this material, you can draw it into these incredibly stiff fibers as well, which is different than most glues. They're not that you wouldn't get that sort of behavior in them. So that was where I got really interested. I said, what's going on here? So we studied some of the kind of the fundamental structure of this. And based on some of the previous studies, we knew that this was going to be based on cellulose. Cellulose is the most abundant biopolymer on the planet. It's what makes up trees and all plants. It's found in certain types of bacteria. They can produce it. Certain type of fungi, even one type of animal called a tunicate can produce cellulose. So it's a pretty common building block in nature. And if you've probably heard of cellulose as well, but didn't realize that if you are trying to add more fiber to your diet, that's cellulose, right? It's this plant material that our bodies don't digest, but it's also a really amazing building block for materials because what is cellulose? It's these long sugar chains. It's just really, really long chains of simple sugars that then can form into these small crystallites, these tiny little crystals. We're talking about nanometer length scales and very high aspect ratio. So that means they have a small diameter and a long length, and they're incredibly stiff. So when you add them into a material, it gives them mechanical stability. So that's where the mechanical integrity of a tree comes from a lot, is the cellulose that's in there. So these Viscin fibers are actually 50% cellulose. Okay, and so that's interesting because that tells us, well, that's probably where the stiffness comes from. But what's not normal is this processability, the fact that you can just take this thing and stretch it and into a fiber, or also, as you can see in the slide, you can also make films out of these. If you make a fiber, stretch it a little bit, and then pull in a third direction, all of a sudden you form these films, these freestanding films. So it's got an amazing processability, but it's all based on humidity. There's no, like, I'm not heating this thing up. I'm not really doing anything to it except if I keep it humid, it's very pliable and can be pulled into different shapes. But if I dry it, then it becomes very stiff and like it has great properties, right? So we were interested. Well, how the heck does this work? And so yeah, we're now we've explored the structural factors behind that. But in the paper in the paper that I uploaded or that I sent to Katerina, you can kind of read a bit more about that if you're interested. But what we're interested in is now is maybe how we can apply this, in different types of applications. Since it can be formed into films, it can stick to skin. In this paper, we actually explored the idea. Well, could it be used to kind of seal a wound on the skin? And we showed at least on some they got a piece of my student Neil's got a piece of pork from the butcher that still had the skin on it, and he showed that he could seal it. He actually wore a piece of mistletoe on his hand for half a week, and even when he was washing his hands, it stays on there. But then he could peel it off later on. So we wonder if this could be kind of useful for those sorts of applications. Um, and we're kind of still exploring this, and we're exploring kind of the chemistry of adhesion at the moment. But yeah, so that's I find this a really interesting system. I think there's a lot of potential there. But we're much earlier in the stage of our understanding of how the chemistry works and how this process actually works. So if there's any questions on the mistletoe, I'm happy to answer those, or I can proceed to just go to the third system that I'd like to tell you about. But okay, so the last system or the last animal that we're currently looking at in my group is called a velvet worm. The real name is an onychophoran. They are kind of an ancient species or an ancient group of animals. They tend to live in tropical and subtropical environments, kind of in the leaf litter in forests. And they are carnivorous little animals. They actually look kind of like a caterpillar or a cross between a caterpillar or a slug. I had a colleague who saw one of them the other day and said it looked like a slug, but they're actually really cute, whereas I don't think a slug is quite cute. They called velvet worms because their skin actually has this kind of velvety texture to it. They're a really interesting animal for a lot of reasons. So they're really quite ancient. I mean, we're talking about at least 400 million years old in terms of when the first things that you could call a velvet worm were around. And there's only about 200 species that are identified on the planet. We study one that's from Australia, but we're also going to be starting a project on one from the Barbados. And it's a really interesting creature. So it's interesting for a lot of reasons. One, they give live birth, which is, it looks like an insect, but it just gives live birth to little baby onychophorans. But the reason we're interested in it is because it has this really weird hunting strategy. So basically, they are able to fire a projectile slime out of two appendages on their head, known as the slime papillae. Talking about it, I can't do it justice. I think what you have to do if you're really interested in this is there is a David Attenborough narrated video of a velvet worm hunting, and that will give you everything you need to know. It's kind of a slow-mo, super slow motion video of the velvet worm firing its projectile slime. It's a lot of stuff, a lot of goo that comes out of the face of this thing. So why did I get interested in this? Well, I met a guy who came to give a talk at our institute, who was interested in the ecology and life cycle of these animals. And I started chatting with him about it, and he said he had a PhD student who was interested to study this slime, and then he told me a little bit more about the slime, and I was really hooked. So what happens is that when it shoots the slime out, it's a fluid. Okay, it's a liquid. It's stored inside the body as a liquid, and as it leaves the slime papillae, it transitions into this gel-like phase. So it becomes a solid in midair. It's what we call a viscoelastic solid, but it's kind of like a jelly, silly putty-like material in midair. Then, as it lands on the prey, so the prey being typically other insect, or not other insects, but actually insects. When we have them in our lab, we feed them crickets. They entrap them with this slime, and the slime dries and hardens and captures the animal. And so then the velvet worm will go and basically just eat the animal, or sorry, eat the insect just right there. And so we're interested, though, from the perspective of looking at this slime and saying, okay, here is a fluid that is rapidly transitioning into a solid, and we studied the fibers, and the fibers again have a stiffness that's comparable to nylon. Okay, so what's interesting is that if you let the velvet worm shoot the slime and you collect it in a tube, so an Eppendorf tube, and you let it sit there for just a little bit, it will transition. As long as it doesn't dry, it'll relax back from that gel state back into the fluid state, right? So it becomes basically a liquid again. And all you have to do is let it just kind of relax for a little bit without agitating it. But what's really interesting is now that you have the slime outside of the body, you can use kind of mechanical shear. So I just mean, you stir it up a little bit, and then you pull it. Like if you take a pair of tweezers and you stir it up and you pull it away, you can form those fibers again. Okay, so outside of the body, you can form these fibers. That means that all of the ingredients that are necessary to form these fibers are in the slime. And that means, as a biochemist, as a chemist and a material scientist, I can use all the tools that I have to investigate how that works. And so that's what we did with some slime that we got from our collaborator. And it's really, really interesting, because what we found out was, okay, we can form these fibers, which again have properties that are comparable, let's say, to nylon, at least in terms of stiffness. We can take that fiber, we just put it in distilled water, or just water, and it will dissolve again. Okay, so it's like recyclable just using water, but then we can take that dissolved fiber solution and we can make another fiber. So it's fully recyclable. And so that's when I was like really hooked on this. So we started looking into this, and we made some really interesting discoveries in terms of the composition of the slime, in terms of what is actually changing chemically and biochemically at the nanoscale and at higher length scales. And it's, again, this one is really based on proteins. So the muscle viscus also made on proteins. Mistletoe Viscin is made of cellulose, but this one is made of proteins, and the proteins are what we would call mechano-responsive. So that means they respond to mechanical forces. So when we shear the slime or when we pull the slime, the proteins are actually changing their structure. They're responding to these mechanical forces. They're changing their structure, and this is causing them to kind of want to stick together, and that's what allows these fibers to form, or at least that's what we understand so far. But again, I have to say we're not we still have a ton of work to do to figure out how this one is. We've got some constantly getting new results that make us completely turn around our our model and our hypotheses, and readjust. But for me, that's I don't find that frustrating. I find that the fun part of everything is kind of having to think about this a little bit more deeply, and look at it more fundamentally, and try to figure this out. So um, that's kind of just a brief overview of those three systems. Again, I don't want to take too much of anybody's time, but I'm definitely open to talk further about any of the things that you find interesting, um, or answer other related questions. So I guess that's it from me now, and I'll open it up for everyone else.
Well, thank you so much for this really interesting research you shared with us. And it's so cool that you can like completely recycle it so easily. That's so interesting. So you told us how you came about to, you know, why looking into mistletoe. So is that like are you planning or to go, is there a way to go systematically through like organisms and figure out what could be useful, or is it always like some, you could use some algorithm or AI driven, or is it you think like this has to be human based, like kind of having more insight, like you said in the beginning,
Yeah.
a broader range of knowledge?
Yeah, I mean, I think, well, you asked like so many so many good and important questions right there. The funny thing is that so much of the work in the field of bio-inspired materials is focused on such a small number of materials. And it's almost like history that has done this. I mean, people started studying one thing, and then a bunch of other people came into it, and let's put it this way: the more that's already understood about the system, the easier it is to kind of jump in and make kind of deeper discoveries, um, or more meaningful or more, let's say, impactful discoveries, where like what will influence something that's already going on. Whereas when you start with a new system, you're kind of starting from scratch, which I like, because basically anything you find out is new. Um, so there's that aspect of it. To go through it in a systematic way, in terms, I guess, of finding new model systems to look at. There's so much diversity in nature. I don't know of a good systematic way. Actually, a lot of the really good work on this, on the diversity in nature and the types of materials that are produced, it was done in like the 1700s and the 1800s, when they were discovering these animals. When we were looking at mistletoe, we're citing papers from like the 1820s. Even back in the time of the Greeks and Romans, there's I think we have a citation from Pliny. It's kind of crazy, because that's where like the really, there was this real interest in just understanding nature for the sake of understanding nature, and for documenting the variety and diversity of different creatures on the planet. Now you'd be hard pressed to get like a nice publication out of something like that, but that wasn't the motivation back then, I guess. So if you actually dig into this old literature, you can make some really interesting discoveries, which might lead you to a new model system, to a new interesting animal or material. And actually, I'm up for sabbatical next year, and that's kind of my goal, is to kind of dig through some of this old literature, to maybe take a trip to the ocean, to the beach, and actually just walk along and try to find some new things to focus my work on. I mean, we are starting a new project on sea cucumbers that we haven't published anything on yet, but this is also a very interesting animal and a very interesting material that they have, their outer wall, their outer skin wall. So I guess to answer your question, I think it does take, there's not a way to automate this, but so it does take some scientists looking, maybe, in my opinion, looking more deeply. I mean, this is organismal biology now. It's no longer chemistry. It's no longer material science. You really got to look into these old literature and try to pick out some interesting model systems that way. That would be at least my thought.
Yeah, I love that you said that, because we scaled research in general, but we do all these applied projects, and you have to write it that way for a grant, and so and I don't know. I know from even Nobel laureate speeches. They came to like a foundation in Portugal to speak like once a month or so. Anyways, most of them said, don't do research that you think will be useful, just do good research out of
Yeah,
curiosity. And this is happening less and less, I feel like, and it gets also overcrowded, and I kind of get like really anxious because we need more innovations, but
yeah,
where should they come from if we have like a strategy that, because we think we're so smart, right? But we are
sure,
not. So
sure.
it
No,
makes me.
I mean, yeah, it is a sad aspect of kind of the way current research and publications work, that this kind of fundamental research, which is so important, that is not driven by a goal of some application right away, because we don't know what benefit we might gain from understanding these systems and this natural biodiversity more. We can't say at the outset that if I do this, that it's going to lead to some sort of product in the future. But you never know. And actually, just, I mean, personally, the thing that motivates me is really this sense of curiosity to understand these things, to kind of figure out these problems. And I agree with you that it is a shame that there's not more uh, funding for these sorts of things. Now, in Canada, there is still what's called the Discovery Grants, where it is meant to fund fundamental science. And in Germany, while I was there, you had the DFG was often funding fundamental science and research that was an interesting research question. It didn't necessarily have to have a clear application that was going to come out at the end of it. But it's true that this is getting less and less, this sort of funding, and it's often going to health-related research, and other things like that. So yeah, I would join the call to say let's keep funding fundamental research, because again, in talking about the muscle byssus research, this discovery of DOPA as a possible for surgical adhesives, that wasn't the intention at the beginning. It was simply just somebody, I know my former PhD supervisor, Herb Waite, looking at these muscles and saying, how do they do that? That's so crazy. And having the tools and the knowledge of how to ask that question and try to answer that question.
Yeah, I agree. And somewhere is the key for probably all the problems we have. We just don't, we just don't know enough.
Yeah,
So, and we don't know where to look because we don't know even, we
yeah,
don't know anything. So,
yeah.
anyways, I want to give also others opportunity to speak. I will ask maybe if there's time later something. But Kirko, Frank, Guter Dr. Shah. Yeah, Dr. Shah, go ahead.
Yeah, first of all, thank you so much, Matthew, for sharing your wonderful work with us. And my question is kind of thinking about the biomimetic nano platform, as well as the, for example, when we are thinking about microorganism related with that, also some of the features that we need to get into consideration. For example, thermal effect, and it can be even photothermal effect, and some of the heterostructures. I was just wondering, because you just work on three different kind of things, did you find any, I mean, interesting things around that during your work or not?
So you mean photothermal properties of these materials?
It can be that one, but my question mostly is related to the nano platform and how we can just merge your finding somehow with other things in related with the microorganism if we want to just produce a new thing.
So when you say, sorry, you mean microorganisms like bacteria,
Can
or?
be macrophages specifically.
I see, I see. I would say that's probably out of my realm of expertise. Although I do have to say, at least in a lot of the marine organisms that the ones I've studied, but also knowing the field pretty well, a lot of them um, do appear to have um, antimicrobial surface chemistry. So a lot of them contain halogens, so like bromine, iodine, and chlorine element in the surface of their material and nowhere else. And nobody really understands why they do that. And one of the hypotheses out there is that this is related to kind of antimicrobial activity, so that these materials are not degraded right away in their environments. And also, I can say that the velvet worm slime also contains very small, short proteins in there that have no known function except to possibly be antimicrobial. So I think there are a lot of obviously the first antibiotics were discovered um, produced by fungal spores. So I think there's also definitely aspects of this. It's coming from a material perspective, it's kind of one of the things that I don't often pay as much attention to, but it's definitely there. I think for someone to look into. But yeah, no. I have to say I'm not like a real expert in that area, but I think there's probably some potential along those lines.
Also, did you find any kind of response for example, hypothermic effect or not?
Not really, but what's interesting, muscles, we study a type of muscle called the blue muscle, middleus edulis. It lives in, um, it's the one that you typically find at restaurants and that sort of thing, or at the grocery store, and it lives in the marine environment, right on the rocks where the waves are crashing. But there's other types of muscle. I mean, there's so many other types of muscles, and we actually once studied some threads from mussels that live in the hydrothermal vents. And we never really went far with that study, but we were really interested in what does that environment, which is going to be very high pressure, very different temperatures, and being close to a hydrothermal vent, where there's probably a lot of um, sulfur in the water and these sorts of things, how that might affect the materials. But in terms of understanding kind of the thermal properties of any of these materials, not too much that I can think of. But yeah, it could be an interesting comparative study to think about. Could you look at these organisms that live in very different environments in terms of temperature, pressure, and see how they have evolved differently?
And amphipathic can be another one,
Yeah.
how it works in the lipid service. So thank you so much.
Yeah, thank you.
I guess I'll jump in here real quick. Matthew, quick question, or Dr. Harrington, are you for? I'm pretty sure you're probably familiar with Dr. John Wilkers on
Yes, of course. Yeah.
at Purdue, right? Are you
Yeah,
guys
yeah,
like,
yeah, yeah. I know him.
yeah, yeah? Because I was like, as soon as he said muscle
Yeah.
together.
Sorry, I missed the question.
Maya, you're cutting out. We can't hear you. We didn't hear anything you said.
To Dr. Harrington, right?
We didn't hear anything you said, Mayor. Could you repeat?
Oh, okay, okay, all right. So I was saying, Dr. Harrington, have you like connected with John Wilkers? Like, are you guys connected? And
Yeah, yeah. So I see him often at conferences, and actually, it's funny. We found out that his sister lives on the same street as my parents randomly. So I mean, I've definitely connected with him, and we've talked about this quite a lot, and have discussed these things. So on our latest paper on the muscle adhesion, he was the one who wrote the perspective, or the kind of the introduction to the paper in the journal. So we definitely connect. We have never collaborated, and but it's, I mean, that's pretty normal when you're in this sort of field. But he's also doing absolutely fantastic work on also on adapting some of this um, chemistry to make synthetic polymers. And I know they're doing a lot of really cool work on, because actually I saw him present at the Adhesion Society conference a couple of years ago, and they're developing kind of muscle-inspired, synthetic glues based on a lot of this chemistry as well. Yeah, so he's doing fantastic work.
yeah, yeah. Actually, me and him like I connected with him, and he sent me samples. We were trying to like, because I have the eyelash extension business too, right?
Ah, cool.
So I was trying to use the muscle, basically the polymer there, as a potential adhesive to have a bio, you know, uh, class adhesive instead
Yep.
of CA, right? But when I
Nice.
got it, but as a polymer, right, the problem was it was like it cures ultra fast. It was like boom, as soon as it hits,
Yep.
it was super fast. So the curing process needs to be a little slower. So, but on the like you got to mix bag of tech here that sounds very interesting. One thing that we do uh, in another room is I think it'd be kind of cool to bring your tech up and then have a session with a bunch of like entrepreneurs and other technologists of how like identifying opportunities for this stuff, because like the fibers you're talking about, I was like, man, paintbrushes and
Yeah.
bristles and all types of stuff is there.
Sure.
You know what
Sure.
I'm saying?
Yeah. Sure.
Right. I mean, I'd like to know more about the adhesion stuff, because ultimately it's like, we're looking for an adhesive that is cures a little slower, right? Not instant,
I see.
because you got to place the lash extension on the lash and stuff,
Ah, I
right?
see. Yeah. Yeah. Yeah.
Yeah. But even the fibers you got, like the fibers you got there, I was like, wait, can we manufacture the fibers and they're sticky? You can just place them on there. Like, there's a lot of cool stuff. I think if you can come, me and Katerina are good friends here. I think we can, if you'd like, we can do a room on extracting all the potential opportunities from this and get a bunch of other minds to see what opportunities there are from your tech. What do you think of that?
Yeah, I mean, it's an interesting idea. Like I mean, I think a lot of what we're doing is fundamental, but there's definitely these possibilities, which we're definitely we're looking into these ideas as well, and we're interested definitely in seeing what can be possible. I think from maybe I'm slightly cautious, but I think from my perspective, I want to understand these new systems. At least I want to understand them a lot more um, fundamentally before I think we can really start mimicking these things. But that was also the case with the DOPA system early on, they didn't fully understand it. They still don't fully understand it, but they were still able to make synthetic materials that had really interesting properties, just based on a partial understanding. So yeah, I think for sure that could be really interesting.
Yeah, it's more of a, hey,
Yeah,
what can be
let's move
produced?
on to the because we have a lot of people on.
Sure,
Oh
sure.
yeah, sorry.
So thank you. Frank, Joyce, David, Bill, Dr. Heidi, go ahead.
I see Dr. Heidi flashing mics.
Yeah, I thought I may share my research since 2008 on biomimicry. In the Congress of Entomology, I had some early research in biomimicry of termites, the white ants, and how we can actually mimic termites' mounds.
Yeah.
And in terms of temperature, so I thought that because you've been asking about the temperature, and another application for biomimicry from different angle, which is trying to mimic the mounds and actually have the same scale of the holes they create in mounds, to keep the temperature inside the mounds at 27 degrees all time. And there is a building in Kenya, Eastgate Harare, which is being built with the same structure, so different scaling, of course, and the water piping as well. So it gives actually two solutions to the water pipes and the canals created by those ecosystem engineers, termites. The other research which I've been working on as well, medical leeches having an anticoagulant biological matter natural, which is antiheparin. The coagulation of the blood, we use heparin. And the heparin, to be synthetic in labs, it's really expensive, really, really expensive. However, we have it in nature in the medical leeches, which is again grow up by in some areas in Africa by the canals. So those are the two areas I've been working on in the last 20 years. So
Wow.
biomimicry, it's really inspiring us to go forward in many solutions and in many ways. But I don't know why this science, it's growing very, very, very slow. However, it has lots of answers for many problems. But I can tell that there will be a golden era for using all those green, especially talking about the COP27 and the climate change. There is no way to go around it without having those green solutions around. So this is my share because I would like to learn more and listen to you more. This is Heidi, and I'm done. I'm complete. Thank you.
Yeah, that's really interesting. Thanks for sharing that. That was really cool.
Guta, the question you texted me in the back channel. Please go ahead.
Hi, good evening, everybody. I have two questions, but they would be correlated depending how things would be done or work. The first one occurred to me when you mentioned that the velvet worm does live birth. So immediately brought me to ethical treatment of animals, right? How this is done, and the concern, and of course you probably have a very good answer for us. But I think it's important to touch this point. And the other one is in terms of scale production, is it viable from so many? I mean, are you going to use the animals? Are you going to reproduce them chemically? Or if you are, then how are you going to still clean and sustainable?
Yeah.
Right?
No, that's a
The
very good question.
architecture interior industry, 90% of what we have to work with is either harmful for our health or to the environment, or both. Thank
Yeah,
you.
absolutely. No, I think you raise two good points there. I mean, in terms of the velvet worm animals, I mean, we, you know, I mean, I'm speaking more because we don't so much have very many in our lab. At the moment, we were mainly using, we were getting slime from our collaborators in Germany who had gone for collection. Well, they go on collection trips and they come back with maybe 10 to 20 specimens that they raise in the lab. And you are able to collect the slime from these animals without harming them. So basically, all you do is you would pick them up with a kind of soft, flexible pair of tweezers, aim their head into an Eppendorf tube, and then when their antenna or the two antenna on the top of their head touch the inside of the tube, they'll think it's a predator or prey, and they'll spray the slime into the tube, and then you put them back in the tank or in the terrarium. So in terms of ethical treatment, I would say they're treated quite okay, like any type of pet. Not that they're a pet, but they are fed and so on. And where you don't have to kill them to take the slime or to get the slime, which we use to investigate. So from that perspective, I think it's okay. Do I think we're going to start an industry of materials that are based on any of these animals? Not really, especially not the velvet worm. I mean, it's simply, you can't upscale it. We're not, and I want to make this very clear. We're not proposing that we're going to use velvet worm slime to make materials, to make a recyclable material. That's obviously not sustainable. But what I was trying to say earlier is that if we can extract fundamental chemical, physical principles of how these work, we can try to mimic this. And why is that? Why could that be sustainable? I don't know if it will be sustainable. Why could it be sustainable? It's possibly because these materials, their chemistry allows them to, if we really understand how this works, we understand why, you can activate this material or activate the slime proteins and cause them to turn into a solid. And then if we put them in water, that they dissociate and form back into a solution. If we can understand that, and that's what my hope is, then we can recreate this in a different system. It's also possible to express proteins recombinantly using bacteria, and that can be upscaled. So you can basically have a large fermenter, and you can produce large amounts, gram quantities of, at least at this point, gram to kilogram quantities of these proteins, which then can be processed into materials. That's what's currently happening right now with spider silk. So artificial spider silk, there's several companies around the world that are upscaling or attempting to upscale production of these proteins using bacteria, and then using those proteins to make fibers, and these fibers to make fabrics, and these fabrics to make clothing. So there's, or at least at one point, there was a dress in the Museum of Modern Art that was made from artificial spider silk produced by this one company. Is it sustainable yet? Is it cost effective yet? Not really. But it doesn't mean it won't be in the future. And I think it is possibly a good avenue for getting around the fact that, well, proteins are actually probably better for making some of these materials than petroleum-based molecules. And bacteria are at least able to make larger amounts than the animals themselves, and more ethically, I would say. So I agree with what you're saying, and this is a constant question that comes up, is like, can we upscale this? And at this point, No, I don't think we can with a lot of these systems, but it doesn't mean that we shouldn't try to understand how we might do that. At least that's my thoughts on that.
Yeah, thank you. I don't know if you know Neri Oxman from MIT.
I definitely know of her. Yeah,
Oh, yeah.
I don't know her
Oh,
personally though.
yeah, I had a meeting with her. She's starting to open a company here in New York City, and she was talking about it's really interesting. And yeah, I think you guys should work together. It's really interesting work because I don't. You probably know of her work then,
Yeah, I
but
do.
she's always looking for like new material, and now she's looking, I think, for the company also to she stopped at MIT to like open a company to do like stuff. It's
Right. Yeah.
really interesting. Maybe you guys should meet one day. I know Kirko and Frank, you had questions. Please go ahead.
Oh yes, yeah, thanks. So these very interesting research. I would like to understand, ask about the muscle, um, the slice number is, sorry, the page 3. So on the page 4. Sorry. So as you mentioned, that this one major advantage of this muscle food, the biopolymer, is that it actually can replace water. It works as glue underwater. So it's different from the other two. So in this, I would like to understand a little bit more of the mechanisms. So there's a few agents here. There's the core. There's the like a liquid crystal,
Yeah.
as you mentioned. So the cross linking, is it by hydrogen bonding, or
Oh yeah.
with the red one coating? Is that
Yeah.
is a kind of what role does it play? And also, for as I understood, I did some search just a moment ago, that the way the fibers stick on the rock, for example, the literature explains this by first somehow wetting the surface with its own, maybe the green ones, I don't know. And then could you go into a little bit
Yeah.
more how
Absolutely.
the force works here? Yeah, thank you.
Yeah, so it's complicated. But yeah, you asked a lot of really good questions. I'll try to unpack it. So this image on page 4 is kind of a schematic of how we see this process, kind of making an analogy to a typical polymer production process. So it's kind of a schematic view of how this process works. And so the Bissell thread actually has 3 parts. It has the plaque, which is the adhesive, the glue we call the plaque. It has the core, which is this fiber that I talked about earlier, which is the Kevlar-like fiber that we have. It's actually based on collagen-like proteins, which is, collagen is a protein that makes up a lot of your body, actually, tendon, and it's in your bones, it's in your skin. And then finally, the cuticle is an outer protective coating. It's an abrasion-resistant yet extensible coating. And so these 3 parts are what make up the thread and what combine together to give it its great properties. Each of these parts is formed from a different group of proteins, and each of these groups of proteins are stored in different types of storage vesicles. Again, these are the little bags filled with fluid proteins. And so during the formation process, these contents of these vesicles are secreted into this very narrow groove that's inside the thread forming organ, the byssus forming organ called the foot. And so what they're doing is they're getting secreted out there, and then they're just kind of self-assembling into this structure in a matter of minutes. And again, you're going from a liquid phase suddenly into a solid phase. So how does that work? Well, that is where the cross-linking comes in. And you asked the question, is it hydrogen bonding? Well, there might be some hydrogen bonding there, but what makes the muscle byssus really special in its cross-linking is that it's based on a very special type of unusual type of crosslink called metal coordination crosslinking. So again, I don't know how much everybody's background in chemistry or basic bonds are, but typically you have these types of bonds called covalent bonds and then non-covalent bonds. Covalent bonds are like essentially permanent bonds between two types of atoms, like a carbon-carbon bond or a carbon-hydrogen bond. These have high strength, but also once they break, they're kind of not reforming. They're not dynamic bonds, as we would say. Then there's non-covalent bonds like hydrogen bonds, van der Waals forces, electrostatic interactions. These are bonds that at least under the marine environment, temperature and pH and everything, they're going to be a lot weaker and what we call transient. So they're constantly like breaking and reforming. Like hydrogen bonds, that's what holds water together. And these are constantly just breaking and reforming and gives it the kind of liquid nature of water, right? Metal coordination bonds are really funny because they sit at this place right in between a covalent bond and a non-covalent bond. They're really strong. They're way, way stronger than a hydrogen bond. Yet they're transient, they're reversible. So if you break a metal coordination bond, and this can be based also on these dopa residues or on another amino acid called histidine. Basically, what's happening there is so oxygen atoms that are on the dopa, for example, they have lone pair electrons. That means they have a couple of free electrons that are sitting there on the oxygen, and they donate both of those electrons into the metal ion. Okay, again, I don't know where everybody's chemistry is, but basically what it's doing is it's donating both of the electrons to form this type of bond. Whereas typically when you have a typical covalent bond, one electron is coming from each atom. So this makes them strong yet reversible. And so these bonds are what is actually being used to trigger that liquid to solid transition. And the cool thing is that they are pH dependent. So all these proteins inside the vesicles are stored at low pH, so it's slightly acidic pH. But then when they're secreted into seawater, which has a pH of 8, this is slightly what we call basic, these bonds can form all of a sudden, right? And so you have this really rapid formation of these metal coordination bonds. So that's what is allowing your kind of self-assembly or your cross-linking. And then in the actual material themselves, these bonds, because of their nature of being strong yet reversible, provide that high toughness that I talked about earlier. And they provide also self-healing behavior. Because if you break these bonds, unlike a normal covalent bond, they will reform when they're kind of brought back together, when the group is brought back together. So you can kind of break it apart, and then it over time will recover back to its native state. You can recover your material properties. So I guess, I mean, you asked a lot of questions at the beginning there. I explained at least, I think, some of them, I hope. Yeah, was there anything I didn't get there?
Yeah, that's great. I mean, it's great to learn that today. I mean, I guess it's like a jackpot. I did the makeup of my a great hole in my knowledge that there's I mean, Mother Nature has is very versatile in terms of linking stuff. There's forces. You
Yep.
mentioned the metal coordination. Yeah. So maybe I'll read up more on that detail of chemistry and physics. Thank you.
Yep. Thanks.
And Dr. Harrington, are you familiar with the
Yeko
research?
was next. I'm
Oh.
sorry.
Oh, okay.
Yeah, I'm gonna make it real quick. So, super cool
Oh,
research you
thanks.
did in there. So I was wondering, like, how does the the transition between like of the material go between like I guess as you go across a given range of humidity. Like if you have like let's say you have like a blob and it's like maybe like a 2-inch blob, and between one end and the other end the humidity changes, will the consistency of the material stay like equal, or will it like kind of have phases of change? And the reason why I
That's
asked this, I was just like finish this real quick. The reason why I asked this is because like I think about like wounds, right?
mm,
If you can have something that's like more glue-like in one place and more solid in another place, you could plug a wound with like a small amount of material. You know what I'm saying? So
yeah.
that's what, yeah.
That's yeah, no, it's a really good point. And I think yeah, that's totally possible. So with the mistletoe, absolutely. You know what's funny about this material is that the humidity from your skin is enough to activate the tackiness, the adhesive. So even just if it's close to in contact with your skin, it's already going to activate that um, to make it more sticky. And I noticed this even the first time I was playing around with it, like when I had it in my kitchen. I cut one of these in half, like a fiber in half, and I held two ends together between my fingers. And again, the humidity from my fingers was enough to activate that adhesive so that it would actually heal the two ends of the fiber together. So it would cause them to stick them together. So your point is, do you have local regions of your material which might have more moisture than another? Is there like a gradient going from, like, if it's on top of a surface that's humid to, a if I understood correctly, it's does it have kind of a gradient of properties going from a humid surface to a dry environment? I think it would for sure, and I think that might be why it does have this kind of weird behavior, that it can stick on your skin. It will be flexible, but it's still quite stiff. Um, yeah, no, it's a really interesting idea. And in terms of plugging wounds, again, we haven't messed around too much with that in a real context. We've just kind of been playing around with whatever we can find at the butcher, because my student wanted to see if he could heal one of his own cuts, and I said no, you can't do that. But yeah, no, I think it's a really interesting question.
Last thing I'm gonna say, Matthew, super cool. If it's possible, I'm gonna talk to some people. I'm in the military, and like
Oh,
from a military perspective, if the medics that are in the field have something that can just stop the bleeding, that would save so many lives.
yeah,
You know what I'm
sure.
saying?
Yeah,
So I'm gonna talk to some people, and I'm gonna add you. I'm gonna follow you actually. And if I can like put pieces together, you know what I'm saying, I'll let you know like
sure.
what I can figure out.
Sure, yeah. Thanks.
Perfect. Denise, Joyce, David, Daniel, Ahmed, you didn't have a chance yet to ask a question, so flash your microphone if you want to ask them.
I just came pretty late. I was thinking of a regenerative use, if you could use some kind of resin in 3D printing to make molds for parts made out of other materials, and then, like you said, with water, it dissolves and you can use it again. That's just a lot of people have had rooms on regenerative things, so I was just thinking that sounds pretty uh, recyclable, regenerable.
Mm-hmm. So can you explain that again? So you're doing on 3D printing, you want to have like a
Well,
something that holds on.
well, you were talking about basically material science. I'm
Yeah.
thinking of material science. Think of it like a film that you print from that is
Yep.
the same thing as this saliva.
I understand. Yeah. Yeah. Okay.
And then at the later stage, when you're done with, say, you mold an aluminum, ceramic, crazy ass part with some other material, I don't
Yep.
care. You use that mold. I just don't know the temperature ranges
Yeah. Yeah.
or
Yeah.
or
Yeah.
how the whole thing works. So anyway, it
Yeah.
was just an idea. I said I got here late, so sorry about
Yeah.
that.
No worries. It's a cool idea though. I like it.
Denise, Daniel, and Ahmed, Joyce, did you have a question? Daniel,
Yeah, hi.
sure.
Yeah, enjoying a lot the conversation. I also got kind of late, but the PDF has a lot of very good information. Regarding fibers for clothing, uh, so I work with alpaca, and
Mm-hmm.
alpaca has these pockets of air inside the fiber. So the
Mm-hmm.
thermal properties of
Mm-hmm.
of a wool are there because it traps air in between the fibers. If
Yep.
you add air inside the fibers, then you have more thermal protection. So to the
Yep.
point that polar bears have like almost like a filament, I mean a total hole in the core. It's
Mm-hmm.
like a yeah. Um, are you aware of somebody doing that research, trying to come up with synthetic fibers that are hollow inside?
I'm not. I didn't know too much about that. That sounds really interesting. No, I don't. I don't know anybody that is working on that though. It sounds like it would be
Interesting.
would be really
Yeah, yeah,
interesting.
yeah.
Yeah.
I mean, alpaca fiber also is antimicrobial. I don't know exactly
Uh-huh.
why, but
Why? Yeah. Yeah.
yeah, but I mean, it works. I mean, you can use your alpaca socks for 5 days and not stinky, not sticky feet.
Oh, really?
Yeah, yeah.
I
It's
did not know
something that,
that. That's really
yeah,
cool.
yeah, yeah. Okay,
I need
awesome. No,
that too.
That's
Just
really cool.
here for vascularization, if you can come up with tubes that
Yeah.
let still some stuff through, like ions
Yeah.
and stuff. If you ever come across something like that, let me know. Like,
Very
no,
cool.
for growing like 3D printing organs and stuff, like
Yeah.
doing really good vascularization that kind
Mm-hmm.
of withstand stuff is still a problem.
A big
So if you
challenge.
come up,
Yeah. Yeah. Yeah.
but it's interesting that Daniel and I have similar requests material.
Yeah.
there we
Yeah.
go.
Exactly.
We should collaborate more. Yeah.
Alpaca. That's cool. I didn't know about that. I see. I'm learning a lot of stuff too. That's good.
Ahmed, did you have a question?
Yes, indeed. Hello, doctor. Nice meeting you.
Hello. Nice to meet you too.
Um, so, I'm very sorry for not catching up earlier,
No worries.
but I have quite the question here. From what I sensed so far, this is material that is regenerative in nature, and I guess it's a biochemical. It's not cellular, right?
No. No cells.
Okay.
I
So,
don't know. I don't know if I'd call it. Well, sorry. Which material you're talking about? The velvet worm or?
yeah, yeah.
Okay. Yeah. No, there's no cells in there. And it's, I guess you could say it's regenerative in the sense that basically what happens is you can form the fiber, then you can dissolve the fiber, and then you can take the solution of the dissolved fiber and make another fiber. So yeah, but it's not like self-regenerative.
Hmm.
You know, you have to get in there and do the work. Yeah, sorry.
Yeah.
Please
So,
continue.
so So does it have? I think hence that it has self-assembling features as well.
Yes, I think it must.
Okay.
Yeah, we don't fully understand that yet, but it must.
So there's something here that I needed to. Here's my question. So I just wanted to confirm the information first. So in a way, can we maybe like have it memory shape itself, like kind of shape something on its own?
So you're asking, can
Can it have a
it
geometric
self-assemble
form?
into a?
Yeah. Can it
Yeah,
have?
I mean these tend to form fibers and fibers only, as far as I know. Um, or I mean, like as far as we've experienced, like that seems to be the tendency, or a gel, like a big blob of gel. But basically, the way you get it to form is you kind of stir up the slime, and then you pull away from that. If you just stick it in and you pull it away, you don't really get a fiber. You actually have to have that shearing first, so it needs to be kind of activated, I think, and then you pull it. And when you pull it, you're going to form a fiber. It's just kind of the nature of that, you know, tensile force on there. It's just going to end up as a fiber. The mistletoe system, which maybe you didn't hear about earlier, that's a system where you can form fibers, or you can form films, or you can, do some other types of different geometries. If you take some fibers and you can actually kind of weld them together with using changing humidity. So I think it's not really possible to get different shapes at the moment. It's really mostly fibers.
I see.
So
Thank you.
who does slime molds? I don't know, Matthew and Ahmed, if you know about slime molds that they can kind of recreate a map of Manhattan, or
Yeah,
make like efficient maps from
I have
like
seen
how they would
stuff like that.
design the
Yeah,
street infrastructure, that it would
yeah,
be better than than how you would
yeah,
design.
yeah. I actually saw. I was at a conference recently and did see some people who were using fungi, and they were using it as so they were 3D printing materials that contained fungal spores or something like that would then be activated, and they would start making a material within the 3D printed material that would then take the shape of whatever material that was. It was really neat. It was a group in Switzerland in Zurich, was doing this stuff. But yeah, I don't know. It sounds really interesting though.
I think it has to be biological in order for that to happen.
Mm,
Maybe you have it like maybe rewarded, like
mm,
maybe draw the map with whatever kind of media it needs to
yeah.
grow, and it would form then that shape or builds
Exactly.
upon it.
Yeah. So this is kind of a newer field that seems to be taking off quite a lot. They're calling it living materials. So people are 3D printing materials that have fungi or bacteria, living cells that then can somehow be activated. So I remember seeing a paper last year where they 3D printed some bacteria that would respond to a certain stimuli. Could have been light, I think. And then when they are stimulated by that, when they receive that stimulus, they're triggered to start producing a certain protein, or to have some activity that then changes the material. It changes the material's properties or something like that. I think this is more and more. I've seen a lot of these papers in the last few years, which are just really, really interesting, really exciting area.
Maya, did you have a last
Yeah,
comment or question? Because
yeah.
I
I'm like,
didn't.
I have also a question.
yeah. So
Is there something? Is there something that we know that just by the structure of the surface becomes antimicrobial? Like I'm thinking, shark skin. Is there something on the molecular structure, not the chemical interaction, but the physical structure that makes something not welcome to bacteria that we know?
Uh, yeah, I don't know too much about that field. I think there are certain structures that can be antimicrobial. So I've seen like artificial structure, like synthetic structures where you have a lot of like sharp, small pillars at the nano and micro scale that can impale the bacteria. But in biology, I don't know, to be honest. That's again, it's kind of a little bit outside of my specialty. But who knows? Maybe you could find a really interesting model system where that would be the case.
Thank you. Sorry, Mark.
Yep.
Oh, that's okay. I've just been chomping at the bit and sitting here patiently waiting to like get my little idea out because it's okay. All right, check it out. Check it out. All right, so Dr. Harrington, I don't know if you're familiar with so MIT within the last year they came out with a hydrogel. I put it in the comments.
Okay.
Based on the lobster, right? So the lobster,
Interesting.
yeah, it's pretty super cool. But basically, in essence, it's like Kevlar, but it's like tendon material. Okay, at the end of the day, the best application is tendon material. But what I see with what you've got is, I think it would be really cool for you to maybe reach out to that research team. I posted it in the comments
Yeah.
because if they got the tendon, you got the sticky stuff to take the tendon to stick to the tendon and stick it to the bone. That's kind of what it
Yeah.
looks like is happening right here, dude. You know,
Yeah.
so if it's, I sent you in the back channel a link to the paper there,
I can
but
see, yeah, materials today, lobster belly, tough, stretchy hydro. Yeah, that's interesting. I'll definitely take a look at that actually. Do you know what group it is by chance? I for some reason I can't open it, but
um,
I can follow it later.
yeah, I know that it's at MIT. I don't know the group. I put it, if you look at your back channel, your messages.
Yeah, I'll find it. I'm still
Yeah,
figuring out how Clubhouse works, but
but is that along the right lines of thinking? Like basically, if they got the tendon material, like the stuff you're doing could be the intermediary that
yeah. So
connect.
actually, I mean, what's interesting? I've been calling this Bissell thread, um, like the fiber. I've been calling it a Kevlar, like, but actually, it's made of collagen. Collagen is what makes tendon, so it's actually more similar to a tendon, but actually, it has some chemical differences in comparison to a tendon that makes it uh, tougher than tendon and more stretchy than tendon. So now I'm definitely going to look at this paper and see how similar it is to the properties of what what we make, because basically the muscle has already combined these two things. You have a collagen tendon like fiber that's actually embedded into this adhesive. And I don't know if you're looking at the slide, but on slide 4 you see one thing we show at the interface. I'm always really interested in these interfaces between where the different parts of the byssus actually come in contact, because actually that's a really hard engineering question, is how do you interface between materials that have unlike properties, because there's this effect called modulus mismatch, which basically tells you that if you have two materials that have very different stiffness properties, you tend to have stress and force concentrations at that interface, which is often where you have failure. You can think of like delamination of, or like, I don't know, I'm trying to think of a good example that everybody knows, but basically, whenever you have materials that have dissimilar properties, like also, your tendon bone interface, like you mentioned, or interface between an implant material in your bone, or an implant material that you put in your body, and some soft tissue, you can get a lot of damage there. And so it's always really interesting to look at how nature solves that problem, how nature deals with these challenges. And a lot of times, what they do is they use what are called gradients. So they actually will have a compositional or structural gradient that then leads to a mechanical gradient. And in that way, you really avoid that interface entirely, because you just gradually transition from a really stiff material to a really soft material. And that's kind of what's happening all along the whole fiber here, especially at the interface between the adhesive and the tendon-like fiber material. So I think I definitely see where you're coming from on that. It's a really cool idea, and I definitely will check out that paper to see what they're up to. But the muscle has kind of already has an answer maybe for that question. We just haven't like looked at it close enough yet, in the sense that you're combining you have an interface between these already. If that makes sense.
Yeah, I do. I worked with that. I saw like the different materials and using the muscle adhesive that Dr. John Wilkers sent me. I was trying to fiddle
Yeah.
around with it. I know
Yeah.
exactly.
Exactly.
I know exactly what you're talking about.
Yeah. Yeah.
Match that, but I think like that tendon stuff that MIT has and your stuff with the muscles, maybe you guys could figure out all the way to the bone, because
Yeah.
that's the hard part, the connection, right?
Oh, definitely, absolutely. Yeah, they like I said, these interfaces are one of the biggest challenges. So one of the areas where this is hugely challenging, that's becoming more and more, I don't know, prominence, is our brain-computer interfaces. I mean, this is probably something we'll see develop in our lifetimes. Um, I know that there's that company Neuralink that's trying to do something like this. But then again, you have a very hard computer material like a semiconductor that's coming in contact with one of the softest materials, human brain. And really, one of the big challenges there is not like an electronics question or anything like that, but rather just a material science question. When you have that interface between a soft and a hard material, this is the biggest challenge. So actually, I think there's a lot of areas which are biomedically relevant where by solving that question or by looking at how nature solves that question, we can gain some insights into how biomedical engineers might also solve it. Yep.
Yeah, thank
Yeah.
you
By
so
the
much.
way, a quick
Oh,
comment.
go ahead.
This discussion is very interesting. So by the way, you mentioned the nature, bio system solving the modulus mismatching problem by generating somehow like a gradient.
Mm-hmm.
It's a quite a intriguing. I hope there will be more literature on that. But I might contact you later because I have some of my own work actually address this similar problems with 3D printing by
Oh, cool.
treating the surface of different say hydrophobic hydrophilic surfaces
Mm-hmm.
by say just factory like common like surface treatment, say plasma and others.
Mm-hmm. Mm-hmm.
And I can actually, I mean, by engineering, we are able to build that into, regular 3D printers so that you can actually have, say, PP, and other
Mm-hmm.
hydrophilic PLA
Mm-hmm.
polymers
Mm-hmm. Mm-hmm.
sticking together. Yeah, just quickly adding that
Yeah, very
piece.
cool.
So, yeah, Katerina.
Oh, yeah, thank you. No, I wanted to, give Matthew a chance to go to sleep. It's
Yeah.
getting late here.
No, I appreciate
I
it.
think you're in the same time zone, so
Yeah, yeah. It's
you're
getting a little
probably
late.
exhausted. You talked for like almost 2 hours, and
Yeah, yeah. No, it's all
maybe
right.
you'll come back.
It was a pleasure. It was really, really
Yeah,
a great experience, and I really appreciated all the great questions and just the opportunity to chat with everyone about this. It was really cool.
yeah. No, we enjoyed it a lot, and it was really interesting. And let's stay in touch. I want. I want to. No, but it would be really interesting to follow your research more
Cool.
and what comes out of your sabbatical from
Yes.
doing like really exploring, going back to the roots of biology and exploring like
This is what I'm hoping for, yeah, and come up with a few new cool ideas that'll keep me going, keep me interested. Yeah,
congrats
sounds
to
great.
have that opportunity, and maybe you can take us along and come sometimes here and tell.
Sure.
That would be cool.
Sure. Fantastic.
Okay.
All right.
Thank everyone also for asking questions, and thank you, Matthew. Have a great night. And we have a room tomorrow with Dr. Onen from MIT, and he will be talking about these artificial neurons that are around 10,000 times faster than regular brain neurons. So let's see how he managed to do that tomorrow. Okay.
Okay. See you later. Thank you. Bye.
Thank you so
Thank
much. Bye,
you.
everyone.
Thank you, Matthew and
You're
Katarina.
welcome. Thank you.
We'll close the room in 3, 2, 1. Bye, everyone.
Bye.